So why is it always in a 5', 3' direction? That means the new strand. Why do we always the newly synthesized strand? Okay, why is it always 5', 3', 5', 3'? Okay, now this is the template, right? Okay, template is here. I just look at it in a linear way only. Okay, so first, is sometimes they will ask why you need a primer. Why you need a primer in DNA replication, but not in transcription. Do you know why?
I'm gonna be really honest. I don't really remember what transcription is.
Oh, okay. Both also use DNA as a template, right?
Yeah.
This one also use DNA as a template. Okay. But here you need to add primer first. Okay. So you're actually adding RNA primers. Correct? Then only you continue to add. So this is the 3' OH. This is the 5' N. Okay, so you add the primer, using the primase. Then only you can continue to add the rest of the complementary nucleotide this way. All right. Okay, because why? Because your DNA pole need an existing 3' OH to elongate.
This is replication, yeah.
Yes, the new strand. You use DNA pole. But if this is DNA template, and if you are using RNA pole, does not need. So RNA pole will straight away add from 5' to 3'. Okay? Yeah? So does not need existing 3' OH. So that's why here in DNA replication, you need the RNA pole called primase. So primase is actually an RNA pole. So the primase add first the RNA primer. Then continue.
Okay, then what's about the DNA pole?
The DNA pole will add DNA nucleotide. Here will be the DNTPs. Initially is
RNA.
here is they use the RTPs.
So the RNA will provide.
RNA, the primer is RNA first. That's why you have to take out the RNA, remember?
So the RNA primer will provide
Go
the
first.
OH group.
Yes. You understand now?
Mm.
Okay. Why do you need to have that additional step? Okay. RNA pole, no need one. RNA pole straight away, nothing there, right? They can straight away add. Okay. But in DNA cannot. So that's why they need to put the RNA first as a short primer. Then only the DNA pole will add from the 3' OH of the RNA primer. Okay, then remember after that, right, they have to remove, isn't it? After that, they have to remove. So here, all in already, right? Then there's a gap here because they have to remove the RNA primer. You see or not? It's because of the behavior of the DNA pole. Okay, so why does it grow in a 3' direction? Okay, so it's because, one, we use DNA pole, right? What's the function of DNA pole?
To what do you call it? Insert to add the
Pole, polymerase. Right.
to add the nucleotides.
Okay, add and join, right? Okay, so here, if this one is an A, the one with the T will come lah. Here is a G, the C will come. Okay, but they need what now? What do they need? They always need the OH group, and then only they join. So this side here is the 5' end. Okay, so they are joining from here. They join one more here, and then this one provide the OH, then the next one join here. This one provide OH, then the next one join here. So this one is fixed already, right? Now the one next one add here, next one add here, next one add here, next one add here. So it looks as if it is moving from 5' to 3'. Do you see or not? So why is like that is because of the behavior of the DNA pole. All right, the DNA pole active site, because this is an enzyme. So the active site must look for a 3' OH. Otherwise cannot bind. So it binds, the active site actually recognize and then bind. So that's why we use the word recognize and bind to existing 3' OH on the new strand. Recognize means what? It is the shape. Recognize and bind, attach. Okay, after that, the DNA pole do what? Will catalyze the formation of what bond?
Oh my god. Phosphodiester.
Yes, good. Phosphodiester bond. So here, so between one and the other, that is how they join them together, right? To join adjacent nucleotides. So that's why they match and then form the bond, and then after that they match, form the bond, they match, form the bond. So that's why it grows in a 5', 3' direction.
Is it important to say catalyzers?
Yes, after the word enzyme is always catalyzed.
So it's an enzyme.
DNA polymerase
Oh yeah. Okay.
is an enzyme.
Okay. Okay.
Okay, so DNA pole is the one that match the complementary base pair. So they make sure that the correct nucleotide comes in to do the CBP, complementary base pairing. Okay, not? Yeah, so they match like this, complementary base pair, and then after that, the phosphodiester bond. So all this is done by the active site of the enzyme. Okay, boleh? And then, okay, so sometimes they'll ask you, okay, the primer. Then what will remove the primer? Which enzyme remove the primer?
Primase.
Huh? What?
Primase.
Primase? No, primase form, you won't remove. Another DNA pole. When we say pole means actually short for polymerase. So there are many types of polymerases. Okay, you only need to remember DNA pole. You're not required to remember pole 1, pole 2, pole 3 unless your school expects it. In your notes, do you have pole 1, pole 2, pole 3 and all that? Pole 1 and 3?
I don't remember.
Okay, so we use the term another.
Okay.
Another. That means it's a different one to differentiate from the one that catalyzed the phosphodiester bond. Okay, so that's why we say another. Will proofread, nah?
Yes.
Okay, proofread means what? They will go and check. So the last stage is where they will check, right? So when they check and found that there is RNA here, they will then remove.
Okay.
Okay, so this is the new one. This is the template. Okay, so then this one will come and then remove. So you're only left with this. So there's a hole here. Okay, now I will extend. So if here there is one strand and then here the primer removed, the Okazaki fragments means that previously there was another fragments here, right? So this one extend a bit and then there is another one here. Okay, Okazaki fragment. So OK1 or Okazaki fragment 2.
Yeah, but how come it can't just be one RNA primer at the start, then the whole thing is
Okay, yeah, I'll show you later on. Okay, so what happens now is if there are several fragments like this, right? So after they remove the primer, okay, the primer removed, right? So what happened now is they have to now close the gap, right? So they have to replace, okay? So the another, just now we say the another DNA pole, right? Will check, do the proofread and remove the RNA primers. So after they remove, they have to put back, isn't it? And then they replace with DNTPs to make it DNA. So now when they replace, right, where are they going to replace? This was 5 prime, this is 3 prime, this was 5 prime here, 3 prime here. So where are they going to do the replacement? From the 5 prime end or from the 3 prime end?
from the 3 prime.
Yes, excellent. It must always be from the 3 prime end because this side here is the 5 prime end, right? 5 prime end is no use because why? The DNA pole, that means the first one just now, right? Cannot go and join, isn't it? So they have to join again. All DNA pole must join from the 3 prime end. So now what did they put in? They put in here, right? Here. So they have to continue from here, continue until the last one. Continue until the last one, there's a gap there. So that gap must be sealed by another enzyme. This one cannot. So what is the last enzyme that will come in?
Ligase.
Yes. So the last one that will come in and join this bond is DNA ligase. Okay, so that's why they say that DNA ligase, the role is what? Okay, so there's so many enzymes here. So DNA pole, first one that we talk about is to catalyze the formation of PDE, phosphodiester bond. Okay, and elongate the new strand 5 to rewrite DNA pole. Then you have another DNA pole, and then DNA ligase, and then you have primase. So all these enzymes, each you have to know. Okay, this one here do what? Catalyze
The formation
of phosphodiester bond.
oh,
Right? This is the one for elongation of the new strand. Okay, from the primer to the 3 prime. Like that. Extend from the primer. Okay. This one here, the another DNA pole is to check, proofread. We call it proofreading. Proofread means to check for mistakes in CBP, complementary base pairing. So it's like either they paired wrongly, right? So they will have to take out and correct it. The other one is in the process of proofreading, they check, hey, how come got RNA here? So they have to remove the RNA primers. Okay, ligase is to join adjacent Okazaki fragments. Okay, how do they join? By forming this part here, the phosphodiester bond. It's actually phosphodiester bond, but this phosphodiester bond is not between two nucleotides. The phosphodiester bond is between one fragment and another fragment. Okay, and primase, this is to add RNA nucleotides to initiate the new strand. Okay, that means to synthesize a short primer. Okay, by synthesizing a short primer, this is to allow the initiation later on. Okay, so in terms of sequence, this is the first step, this is the second step, third step, and fourth step, like that. Okay, all right. I'll explain a little bit more because here they will need you to go and draw the picture, right? Any questions so far before I move on?
no.
Okay, so it's always two strands like this, correct or not? Where will they start to open? Hmm?
Huh? No, what?
Where will they start to separate?
Oh, it's like random.
No, there's a site there called the origin
Oh,
of replication. All
wait,
right.
isn't that just where it starts?
Yeah, it starts from the middle. It does not start from the end. Just now you were saying, why can't it just move from one end to the other end? Okay, because they open up from the middle. Okay, now, so in eukaryotes, you have multiple ORI-R. When you see this ORI-R, that means that's the origin of replication. So this is where they are going to split open up like that, and call this a replication bubble. Okay? Now, it starts from where? It starts from the middle. It does not start from the end. So it's going to start from here and move this side, both sides like that. It's going to open up. This one is going to go this way. This one is going to unwind and open up that way. Like this. This one is called bidirectional. Okay? Now, one of the strand is going to be 5 prime, the other's 3 prime, right? So, okay, I call this 5 prime, 3 prime, 3 prime, 5 prime. Now, so from the middle here, how do you actually label it? So from the middle here, it means that the 5 prime will be coming from here. Correct or not?
Yes.
That means you start to look at one half. That means you don't look at the other half yet. You look at the left half from the middle point. If the top here is 5 prime coming to 3 prime and at the middle, right? So the bottom there must be 5 prime to 3 prime. So here, they will add the primer, and then after that, extend. Agree?
Yes.
So now the bottom one is 3 prime there, right? So here is going to end at 5 prime. So which means that the top here is 3 prime. So my 5 prime will be here. So to initiate, I will put the primer here. And then move towards 3 prime. Agree? Okay. So I'll only look at one half, right? So now what happened is this one is going to unwind further, right? So when it unwinds further, it will become like that. Okay, so just now was stop here, and then the primer was here. This one here, the primer was here just now, right? Okay, just now it ended here, correct or not?
Yeah.
Here, to make it more correct. Okay. Just now was here. Okay, so now the top strand, this is 5 prime, right? Where is the 3 prime? Here.
Yeah.
So how is it going to continue? Do they need to put another primer at the end?
No.
No. So this one will continue because why? There is a 3 prime OH.
Yep.
So you got the end for them to actually continue. All right. You see this? This is the direction of the opening, right? This is the direction of the fork, right? Now, if the strand is same direction as where the fork is going, this is the continuous strand. Okay or not? It continues. So as it open up, there is the 3 prime end. So this side is always 3 prime. So it will continue. As long as the elongation of the new strand follows the direction of the fork is continuous. Now you look at the bottom strand. As it opens up, so now what happens? This is the 5' end, right? Anything there? Nothing there. So if there's nothing there, they have to initiate with the primer first. Okay, not? So that you have the 3' OH, then only it can elongate, but it elongate until there one last gap there. Can not? So it's like that. No? So if this one continue, now I show you the other half, the opposite end. So on the other side on the other side, this is 3'. So this side will be 5', right? If this side is 5', this side will be 3', and this will be 5'. Correct? So now on the 5' end, I will add the primer.
Yes.
Then after that, I will elongate until here and stop. The bottom one, this is 5' here. So it will be 3' here, right? So the top will be 5' here, 3' here. So where would I put the primer? At the origin or at the fork?
The origin.
Yes, perfect. Here. And then here I extend. So now later on, what happened is this one unwind further, right? And just now was what? Just now we continue finish here. Just now we finish here. And then it stops here. This one stops here. Okay, now tell me how to go on from there. Top one or the bottom one?
Or bottom. You
Bottom? Bottom one will do what?
you need 5' to 3'.
This is 3', right?
Yeah.
So what happens here to this end? Continue or put primer?
Primer.
Why?
Because if it's 3' there means the parental strand will be 5'.
No, this is the new strand already. New strand here.
I don't get it. The 3' OH is parental strand or the daughter strand?
The parental one is blue.
No, like for 3' OH, it's to be
We
found on.
always talk about the daughter strand. To elongate from the daughter strand, they need the 3' OH on the
Okay,
daughter
then you
strand, right?
can
Correct or not?
you can just continue.
Yes, just continue. Okay. Because why? This direction is going where? Is it going towards the direction of the fork or away from the fork?
Towards.
Towards the fork. So this one here would be a continuous one. Okay, now the top one.
New.
You need a new primer. Then only they elongate. Okay, so this is discontinuous. So what you notice is if it is going against the direction of the fork, it is always discontinuous. Cannot? So
Okay.
the two halves, so you must know now, always just look at one half will do. All right, the other half will be the opposite because of the anti-parallel. So the way to remember is if the daughter strand is follow same direction as the fork, then it will be continuous. When it goes against the fork, it will be discontinuous. Okay, because there will always be a gap there, and then as long as there's no 3' OH, you have to put in the primer to provide this 3' OH to extend.